For the last video lecture in this week, we're going to cover the final step of cellular respiration, which is the electron transport chain. And on this picture, you'll be able to see it right over on the far right-hand side as the final step of cellular respiration. So to give you a rundown of what we're going to cover right now, we're going to have to talk about redox reactions first. Redox meaning reduction and oxidation reactions. This is the name given to electron transfers. Basically, in one type of reaction, a molecule will add electrons, and in another type of reaction, a molecule will lose electrons. When something is adding electrons, it is reduced, and when a molecule loses electrons, it is oxidized. Also, before we start talking about the actual steps of the electron transport chain, we need to go over the idea that this chain represents electrons falling in their energy state, falling down, as the picture often shows, a set of stairs. Then we will finally move into the 6 steps of the electron transport chain that your book mentions. It's obviously much more complicated than that, but that's where we're going to start. And once we complete talking about the ETC, we'll wrap up discussing how it is the final step of cellular aerobic respiration. This figure is from your book, obviously. We call the reduction and oxidation reactions redox reactions. Often they just work together in a chemical reaction. In the redox reactions, as I mentioned before, keep in mind that oxidation is when a molecule loses electrons. So looking just at oxidation on this chart, we're going to look at glucose. And this is the molecule that loses electrons and hydrogens, essentially, to carbon dioxide. Now, in contrast, we're going to look at oxygen, O2, and in this diagram, it is reduced, meaning it gains electrons and hydrogen atoms that come with it. So one way to remember reduced. An electron has a negative charge. So when a molecule gets more negative charges, its overall charge goes down, right? So say a molecule has a plus sign in front of it. If it gains 1 electron, then it becomes just a regular molecule, like Na+, right? So if you have Na+, it gains 1 electron, then it just becomes Na. Say it gains another electron, so then it becomes Na-. So the overall charge is reduced, and that is just a very, very, very general description. So I've got a few other pictures because I find that this picture in the book isn't the clearest in describing oxidation and reduction reactions, so the next slide has 2 here. I like this top picture because it shows something similar to what happens in a typical chemical reaction, where one molecule will have the electron, and through the process it ends up giving the electron to the other molecule. So this electron donor, it's losing the electron on this side, the electron's gone, is being oxidized because on this side it has fewer electrons than it did in the beginning. So then that means this particular molecule is reduced because here it has fewer electrons than it does here. It gained electrons, so it is reduced. I think this one's a little more clear in the color coding and in the arrows. I also find that this one is effective in illustrating what happens because it differentiates the molecules in an opposing sort of spatial representation. And I like how it incorporates the idea that there is often some intermediary, like say an enzyme, that will facilitate the transfer of electrons. An enzyme or another molecule like what we will be talking about soon here, NADH. So your initial molecule that becomes oxidized starts with some electrons. It goes through a process where it therefore has lost the electrons. It has been oxidized. What we're going to talk about mostly will be NADH, and then once it goes through oxidation, it becomes NAD+. Then for reduction, there is another molecule which will start without the electrons and ends up with them, so that particular molecule has been reduced. So now that you've seen mention of NAD plus and NADH a few times, I want to just give you a little further description on how it plays into the electron transport chain, which is what we'll talk about here in just a moment. I hope you understand that NAD plus obviously has two locations where it can accept an electron, which often comes with a hydrogen molecule, and once it has accepted two electrons, it is then a different molecule. It's got two extra hydrogens. It's called NADH. One electron here, once it binds, it becomes NAD. Once the other electron binds, it becomes NADH. This particular molecule is pretty cool because it can hold high energy electrons. And the electron transport chain takes advantage of the possibility that electrons can be in a high energy state and a low energy state. So what happens is the NADH transfers these high energy electrons to a molecule that is slightly more electronegative than NADH, thereby slightly lowering the energy state of the electron. And then again, the electrons get transferred to another molecule that's a little bit more electronegative, lowering the energy state of the electrons again, until finally they end up with what we've talked about as the most electronegative molecule, which is oxygen. And remember, often electrons transfer with protons, or with a hydrogen atom, therefore making water. That boils down to the idea that oxygen and its strong electronegativity essentially drives and pulls these electrons through the entire process. This ultimate oxidation is, well, reduction of oxygen, but oxidation of the other molecules, is why this is cellular aerobic respiration. It requires the presence of oxygen for this entire process to occur. Now here is your book's diagram of the first few steps of the electron transport chain. What you really need to notice here is that this lighter color on the bottom, this represents the matrix of the mitochondria. That's within the highly folded inner membrane. And then this part here is the space between the inner membrane and the outer membrane of the mitochondria. Also, keep in mind that since this here is the inner membrane, this is where all of these proteins are embedded. Since it's highly folded, that gives a greater surface area for the membrane and a huge amount of space and room to hold many of these proteins that make up the electron transport chain. The most important thing to remember is that what happens here between the space outside of the inner membrane, it's between the two membranes, and then the matrix of the mitochondria, is that there is a gradient. We've talked about diffusion and osmosis and how water and hydrogen similarly will try to cross a membrane from a point of higher concentration to a point of lower concentration. So what happens in this electron transport chain is that as the electrons are moved through each protein, an H+, a proton, is pumped out of the matrix to the space between the membranes of the mitochondria. So that happens 1, 2, 3 different times, creating a gradient where there is a much higher concentration of protons out here than within the matrix. So that creates this almost like a pressure or a strong push by the H+ protons to try and get back to the other side where there's a lower concentration. So we will talk more about that in a little bit when we follow this arrow, but first we'll talk about the actual electrons. So NADH, like we've mentioned before, that's generated with the 2 electrons attached from the citric acid cycle, has these 2 electrons to be donated. What will happen is it'll donate these 2 electrons to the first protein in the chain, thereby becoming NAD+. So it cycles right back. And since these are high energy electrons, this particular molecule accepts them as slightly lower energy. And at each transfer of the electrons down the chain, they lower in energy until they finally reach oxygen. So NAD NAD plus participates here. Remember, FADH2 came into play at the very end, or in the citric acid cycle. It's not in glycolysis, it's not in the other steps, but in the citric acid cycle. It's a molecule very similar to NAD, but it doesn't transfer its electrons to the first protein in the electron transport chain. It actually transfers its electrons to an intermediate molecule here. And this is sort of like a courier, we'll say. Once these electrons from NADH, get transferred here, they're transferred to another molecule, which will give the electrons to this same molecule. We'll call it Q. This is Q. That's short for a quinone molecule that it is named for. So these electrons and these electrons end up going to this. Now, why do you think FADH transfers electrons here? Why doesn't NADH, or why doesn't FADH transfer its electrons here? It's because of the energy level at which it is able to accept electrons. It does accept them. They are relatively high energy, but they're not higher than this molecule. So it's not an easy transfer for FADH to give its electrons to this molecule. That's why it comes into play here. This particular one is at the proper energy state for these electrons to transfer more easily. At this point, there are several electrons that can come into play in the intermediary Q between the first and the second protein of the electron transport chain. This little guy just transfers back and forth within the membrane. So the electrons eventually get transferred to the second protein in the cycle, where again, as the electrons are transferred through each of these, another proton is pumped out of the matrix into the intermembrane space. Intermembrane space. And then a lower energy molecule exists here that will transfer the electrons between these two proteins that we'll call C, Q and C. I guess you don't really have to remember that, it's not in your book, but that's part of the overall process, which will finally transfer the electrons to the final protein, thereby pumping one more H+ ion out of the matrix. And the final acceptor of these electrons is oxygen. This one's finally at the proper energy state to hand the electrons over. So once oxygen gets a hold of them, there are still some H+ ions remaining, but they become bound to oxygen, thereby making water. I'm sure you're interested in the little skull and crossbones here. This is something your book talks about, where cyanide comes into play. Since it mentioned it, and it's in the picture, cyanide essentially binds to this last protein in the electron transport chain and stops up the whole process. So the electrons never make it to oxygen. By basically blocking the step where you generate the most ATP, it's eliminating your ability to generate energy. Now, you do still get 2 ATPs from glycolysis out of every molecule of glucose that you ingest, but that is not enough for the way that your body has developed to sustain metabolism in its existence. So that's just not enough energy. It won't do. That's why glycolysis is sufficient for only bacteria, not a large, very complex organism. That's how cyanide kills you. This is the first step. There are 3 ultimate proteins that transfer electrons to oxygen. The high energy electrons from NADH slowly but surely go down this energy gradient. In the process, H+ is pumped out of the matrix to the intermembrane space, and this next step, this is done. All of these H+s are pumped out to get to the final step where most of your ATP is generated. So this final step here, remember this is the last guy in the electron transport chain, but the final step comes here. This is literally like a mechanical rotor. H+ ions are trying to get back to the other side of this membrane very desperately. What happens is there ends up being a very great difference in the charge, the overall charge of the fluid on this side of the membrane and the fluid on this side of the membrane. Remember, these are H+, so you have more positive charges over here than here, so they try to come together and even things out. What ATP synthase does is it regulates the number of protons that are pumped back through. and it slows it down. It's like, as your book describes, a dam, like the Hoover Dam, right? It's holding back tons of water. And they have a little hole in there with a turbine that spins the turbine. So as the water comes through, that's how electricity is generated at the Hoover Dam. Well, essentially, this is the same thing. This is a turbine, and as each proton or each H plus molecule comes through, it spins this turbine. And as this turbine spins, it's got several proteins attached to the bottom of it here. As they come around, they accept an ADP and a phosphate group. They fit into the protein, and it spins around. Once an H plus is pumped through, that orients the protein in such a way that it's able to connect to the ADP and the phosphate and generate ATP. So for every turn, one ATP is generated. So first, the process of transferring electrons from a high energy state to the lowest possible energy state attachment by attaching them to oxygen generates this proton gradient. Generates one molecule of water and then spins this pump, or this turbine, which is where you actually make your ATP. It's very complicated, and yet simple and elegant at the same time. And don't forget that this is ATP synthase. The name of this whole mechanism here, even though it's several proteins together, synthase, when a word ends in ASE, means it's an enzyme. So the whole function of this is the function of one specific enzyme embedded in the membrane, the inner membrane of a mitochondrion. And I wanted to include this picture so that you can really see where it's at. This is the outer membrane of a mitochondrion, the inner membrane, the matrix space here, and the in-between. So the protons accumulate in this little space, this dark brown space here. And the protons are pumped out of the matrix. And remember, in the matrix is where the citric acid cycle occurs. Now looking back at the big picture, you have a glucose molecule, which comes into play and is initially broken down by glycolysis into two pyruvic acids. NADH is required in this step. These pyruvic acids are then transported into the matrix of the mitochondria, where they're attached to separate 4-carbon molecules, becoming acetyl-CoA. NADH comes into play here as well. NADH removes electrons. Electrons are also removed here. Electrons are also removed here. All of those electrons are then funneled to the electron transport chain that we just talked about. But your glucose, broken into 2 pyruvic acids, are transferred into acetyl-CoA. And then the final 2 carbons are given off. Once they go through the citric acid cycle, you generate 2 ATP here, 2 ATP here, and then approximately through the electron transport chain and the proton pumps and ATP synthase, you get about 28 ATPs here. So that's why this final process is super important. Overall, you'll gain about 32 ATPs in one cell. Now this is an estimate. Books say 36, some books say 38. I think it all depends on the overall process and just how finely you count it, but you can estimate about 32 total ATPs. Now, to give you a larger picture, so far we've only talked about glucose. That's just one sugar molecule. You know that what you eat doesn't just break down into sugar. You eat fats and proteins as well when you eat meat or any number of things. I'd like you to understand that all of your food gets broken down into smaller molecules and smaller molecules, where they are then processed. Some of them will be sent, well, they can all be sent through glycolysis. Some of the fatty acids and amino acids can become part of acetyl-CoA and then transported into the citric acid cycle. But overall, every molecule is broken down. Many of its electrons are taken off and eventually transported to the electron transport chain, where your energy is made. Well, I can't say made, because remember, energy can't be created or destroyed, but transferred. The energy of those electrons from your food molecules are finally transferred to a molecule that you can use in your bodily functions. As a recap, you need to understand the difference and the function of redox reactions, reduction and oxidation. Adding electrons to a molecule means that it is reduced, that molecule has been reduced, its overall charge goes down because it's gaining electrons. And then when a molecule loses electrons, that means it is oxidized. When it is oxidized, generally oxygen is the ultimate acceptor of all electrons, so the action is called oxidation. It has lost electrons, and that particular molecule's charge has gone up. It's become more positive instead of more negative in reduction. The electron transport chain overall is a function of electrons slowly, step by step, moving from a point of high energy to a point of low energy when they finally bind to oxygen. And they are at a point of low energy because oxygen is very, very highly electronegative. This is the final and complete step where all of the electrons are transferred from NAD, NADH, and NAD+ in breaking down your food to generate energy, requiring oxygen, so it is a form of cellular aerobic respiration.